Hello students, as we continue using KnoVi, and we all know that KnoVi is our AI powered educational platform, and this AI helps us to analyze the performance of our students. Okay, from this analysis I wanted to share some really interesting insights from our last test. The platform's AI has done a great job of breaking down our results, showing me a lot of you are finding ribosomes this okay ribosomes a bit tricky. Okay, so in this platform our main goal is to make sure that no one feels left behind. So instead of just moving on the next chapter, we are going to spend some extra time with the topic ribosome. Okay, also KnoVi is so much more than just a tool for checking grades. Okay, it helps me to see exactly where you need to little more help, so I can teach in a way that truly works for you. Let's start our topic ribosome. Let's start our today's class, that is ribosome. Okay, in this slide those are the basic definition and basic information about ribosome. Okay, so ribosomes are essential non-membrane bound organelles found in all living cells, including both prokaryotic and eukaryotic cell, and we all know that they are often called the protein factories because ribosomes are primary sites for protein synthesis. And composition, chemically ribosomes are composed of ribonucleic acid and rRNA. Okay, and also every ribosome consists of two distinct parts, a large subunit and a small subunit. Those two units only come together during the protein synthesis process. Okay, then mainly ribosomes are two types, one is 70s and another is 80s. Okay, 70s mainly found in prokaryotics and 80s are mainly found in eukaryotics. Okay, and then this is our NEET essentials membrane classification. Okay, this is a not direct uh, topic of ribosome, but when we will see that ribosome are non-membrane bound organelles, then there is a question that very much organelles are there without a membrane and some organelles are with double membrane. So this is the classification of without a membrane, membrane with single membrane, and double membrane. Okay, so without a membrane there is ribosome, centriole, centrosome and cytoskeleton. Single membrane হচ্ছে RER, ACR, Golgi apparatus, lysosome and peroxisome. Okay, also vacuole is there, and double membrane we all know that mitochondria, chloroplast and nucleus. So moving on to our next slide, this is the breakdown of 70s. This is the prokaryotic ribosome. 70s has two subunits, one is large 50s and small is 30s, and in our table we can show there is RNA component and also the protein count of the two subunits large and small. Okay, that is 23s 5s for large and 16s for small. Also the protein count for large subunit is 34 proteins and for small is 21 proteins. Okay, then our eukaryotic ribosomes, it also has two subunits, one is large and another one is small. Large is 60s and small is 40s, and also RNA components for large is 28s, 5.8s and 5s, and for small is 18s RNA. And protein count also for large subunit is 45 proteins and for small is 33 proteins. Okay, these two tables are also important for the breakdown of the subunits. Okay, now moving on to our next slide. First of all, the Svedberg unit is the measurement of the sedimentation coefficient and describe how quickly particle settle at the bottom during the ultracentrifugation process, and its value is 10 to the power minus 13 second. One Svedberg unit refers to the 10 to the power minus 13 seconds. Okay, means a very little fraction of time. Then this is the process ultracentrifugation, and in this process we rotate ultracentrifuge at 1 lakhs rpm. Okay, and through this process we can separate very biological samples based on size, shapes and density. Okay, and from our next class, or when we are talking about the techniques chapter, then we vastly study these topics, that is ultracentrifugation. But now we all just know about the what is the Svedberg unit and the value of Svedberg unit. Go on the next slide. So this is our NEET essential three important points. One is catalytic RNA, organelle exception, and dimer formation. Okay. So from the first point, it is the catalytic RNA. Okay. So it is said that 23S rRNA in the 50S subunits. So 50S subunits present in the 70S rRNA. We previously showed that uh, prokaryotic rRNA, which is 70S, has two subunits, large is 50S and small is 30S. So 50S subunit is a part of prokaryotic ribosome. Okay. So it acts as a enzyme which help peptide bond formation during protein synthesis, and it's called that peptidyl transferase. Okay. Then the next point is exception. In our human cell, we are all eukaryotic animal, but in our mitochondria, the ribosome is 70S. We all know that in mitochondria also has ribosomes, but even it is the eukaryotic mitochondria, but it has 70S ribosomes. Okay, and next is dimer formation. That is the dimer formation. If the magnesium ion concentration increases above certain level, which is 0.001 m, and so in this case two ribosomes can join a dimer. Okay, and we can see that in eukaryotic cell, two 8S ribosomes became fused and become 120S units. Okay, actually those calculations are not even like our mathematical calculation. We all know that S refers as Svedberg unit. Now moving on the next slide. Okay, this is the key function of a ribosome. Okay, we all know that ribosome is our protein factory. So the primary function is translation. This is the process of converting the genetic code within mRNA into functional proteins. Okay, then the structural function. Okay, so ribosome serve as a physical platform of mRNA, tRNA, and the amino acids. Then the genetic code interpretations. In this case, small subunit of the ribosome is responsible. Small subunit scanning mRNA and find the start codon, what is the AUG. Moving on the next slide. So this is the catalytic activity and the ribozyme. So ribozymes, the term refers that enzyme with the RNA or the RNA with the enzymatic activity. Also, the ribosome is ensure the codon anticodon formation or the codon anticodon matching, which prevent the genetic mutations. Okay, this is the schematic picture of this large subunit and small subunit, and this is the mRNA with the codons. In the ribosome, the RNA component is not just a structural part. Okay, it actively participates in the chemical reaction of the protein synthesis. It acts as a catalyst. In this reaction, it is catalyzed the formation of peptide bonds between adjacent amino acid in the translation process. Also, it acts as a chain protection. In the large subunit of the ribosomes as a physical tunnel or the channel, which provide a shield. Okay, which provide the shield of the amino acid chain until it long enough to fold and create a 3D structure. You all know that 3D structure cannot be degraded by enzymatic activity, so it's provide a seal or the tunnel. Okay, that's now moving on the next slide. Okay, this is the APE framework of the ribosome. Okay, the ribosome acts as a molecular machine moving along with the mRNA strand, and it always travels to 5' to 3' direction. Okay, at this moves, it shifts tRNA molecule through three sites, that is A, P, and E, or the exit site. Okay, first of all is the A site. This is the acceptor site. It accepts the amino acid tRNA, means a tRNA which carry a single new amino acid. Then the P site, it is the peptidal site. This is the processing center and it is the main site actually. Okay, it holds tRNA that is crucially attached with the growing polypeptide chain. Then the exit site. Okay, this is the outdoor site. Once a tRNA has given up the amino acid to the chain, it becomes empty and discharged from the site. Okay, so this is the A, P and E sites. So this picture I can see is clearly. Okay, this is, we can saw it clearly. Okay, so A site, P site and the E site. So A site is the acceptor site. Okay, acceptor site accept the amino acid tRNA, the tRNA which carries a single amino. Or this is the P site or the processing center. It holds tRNA that is currently attached with the growing polypeptide chain. This is the growing polypeptide chain. Then this is the E site or the exit site. Uh, this is the E site. When tRNA given of the amino acid to the chain, okay, it becomes empty and discharged from this site, from this E site. Okay, so this is the A, P and E model. Okay, so in this model, the ribosome structure, the large subunit, the top part, this contain the A, P and E sites. This diagram also highlight the physical makeup of the ribosomes. The large subunit, this is the large subunit and the top part of where A, P and E sites are located. Then the small subunit, the bottom part that anchor the mRNA. Okay, means the is also present here, also present the mRNA binding site. Okay,